This is Kerri Wachter with Elsevier Global Medical News, reporting from the annual San Antonio Breast Cancer Symposium. Joining me is Dr. Hope Rugo, who is the Director of the Breast Oncology Clinical Trials Program at UCSF and on the Medical Advisory Board of the Oncology Report. Let's talk a little bit about the study that found that there are some biomarkers that may be able to predict trastuzumab resistance and lapatinib response.
Well, there's been a lot of data that patients who have low expression of a tumor suppressor gene called P10 might have resistance to trastuzumab, because P10 is very important for at least part of what trastuzumab does. And again, it's only at least parts. This has been a complicated issue, and there's been a lot of controversy over it. The original study that was published in 2004 was in mice, and human data since then has been conflicting. There's also interest in mutations in something called PI3 kinase, which again is associated with P10 in one of the pathways that is called downstream signaling from the HER2 receptor. And so what Jenny Chang did was look at two sequential, and she comes from Baylor, look at two sequential neoadjuvant trials. What they did first with trastuzumab for a relatively short course of 3 weeks, and then trastuzumab and chemo, and the other with lapatinib for 6 cycles, followed by, again, trastuzumab with chemotherapy, docetaxel in both situations. And what she was looking at really was to understand what biologic markers might predict response or resistance to the two different HER2-directed therapies. And it's actually fascinating data which suggests that decreased P10 would predict resistance to trastuzumab, but in fact, decreased P10 predicted response to lapatinib. On the other hand, a low Ki67 and low MAP kinase that's phosphated, or phospho-MAP kinase it's called, predicted resistance to lapatinib relatively, that blocked proliferation, and may not predict a response to lapatinib. So it's very interesting, actually, that potentially these agents work by different pathways. And in addition, she found that this mutated PI3 kinase also predicted resistance to trastuzumab. So what this means for us is that potentially we could use these markers in the future as a way to individualize HER2-directed therapy for patients. If you have a prediction of resistance to trastuzumab, perhaps you should receive lapatinib. And where we're going to really get the answer to this is from the ongoing neoadjuvant and huge adjuvant trials that are getting tissue from patients, and we'll be able to look at long-term outcome and correlate it to markers that Jenny Chang's lab has developed and tested in these clinical scenarios.
This is Kerri Wachter with Elsevier Global Medical News in San Antonio.